Hi, I'm Ilona Kovács. Welcome you to the Data Visualization and Bioinformatics and Systems Biology course. You are listening to a four-part lecture on human cognition and data visualization. This is the second part of the lecture, and it's about low-level vision. In the previous introductory part of the lecture, we mentioned that low-level vision is mainly concerned by coding for simple but very basic features such as orientation, wavelength or color, luminance contrast, retinal disparity, and movement direction. Remember that it was one of the main messages of part one, that the coding for low-level features is carried out by independent visual pathways in parallel. Without the scary anatomical details, I attempt to show you what these pathways actually entail, although some anatomical detail will have to be shown. This beautiful image shows you the eye and the brain together. It shows the cable-like group of nerve fibers that connect the retina and the lateral geniculate nucleus of the brain. Or it's also called LGN for short. LGN is then sends signals to the primary visual cortex, which is in the posterior or occipital lobe of your brain, which then will have sent its signals in two ways towards the temporal lobe, which is also called the ventral visual pathway, and also towards the parietal lobe of the brain, which is also called the dorsal pathway. Now, these are the most simple things one can say, but let me add that there is some color coding added to this picture. Red actually stands for parvocellular, pathway coming already from the retina, which ends on particular layers of the lateral geniculate nucleus, isolated from the layers of the so-called magnocellular pathway, which is marked with blue. The parvocellular pathway has very little retinal ganglion cells. This is where the name is coming from. Parvo simply means small or midget. And the magnocellular name comes from the fact that it's different retinal receptor cells that are anatomically larger or magnified as compared to the parvocellular ones. Now, the parvo and magno pathways, as we can see, the red and blue lines are being separate up to the primary visual cortex, are isolated. And from V1, from the primary visual cortex, the magnocellular pathway is going to turn into something which we call the dorsal pathway of visual processing. It still seems to keep the blue marking, which means that it actually keeps information separate from the ventral pathway, which is also starting in the primary visual cortex, goes to the temporal lobe and is going to process visual information in a different way than the dorsal or parietal pathway. Now, I promised that we'll try to spare these scary details, but some details we cannot really spare. Let me get my little highlighter. It might help to guide you through this pretty complicated image. It's complicated, but also it is providing you with the essence of low-level visual processing. So it's actually worthwhile to walk through it. Let's look at first the labels in the upper right corner. Here we have color, depth, direction, orientation, and complex form. These would be the basic visual features of low-level visual processing. that we will follow throughout the cortex, and we'll see where and how are those processed in the brain. On the left-hand side, we had different cortical areas that we already mentioned. So we have the primary visual cortex, V1. This is probably the most important one. And it's also the most studied area of the brain. This is something that we share with lower level vertebrates. The primary visual cortex is very similar in other creatures. So we have a great deal of information. There is a very good animal model to study vision at this level. So we know the actual neurons and the properties that code for low-level visual features. We have V2, which is not small, so it actually reaches the other side up here. We have MT in the parietal lobe and V4 in the towards the way to the temporal lobe. Now, these same areas can be found in the upper part of this image, except they are not topologically shown. This is now a map where we can look at the different layers, and within the different layers, we can see what features are processed. The explanation of the two visual pathways starts with the LGN, which was, as I mentioned, a relay station within our brain. It's a subcortical nucleus, which along the visual pathway connects the eye and the brain. In the LGN, we've seen the different layers. We've seen the magno and the parvocellular layers. The magnocellular layers are actually transferring visual information on the direction of motion, and also on retinal disparity. This will give us depth information later on. And that information, motion direction and retinal disparity, ends up on a different layer of the primary visual cortex than information coming from the pinkish parvocellular layers of the LGN. The pinkish layers transfer information on color and orientation that will later on turn into complex form information and actually perceived color. Now, if we follow let's say the magnocellular pathway, we can see that motion direction and retinal disparity information is going to go to the mediotemporal cortex, where that is mainly concerned with actually motion direction, but takes retinal disparity and depth information into account as well. And the simple local motion and depth or retinal disparity information is turned into several maps, 3D maps of our environment. These maps then help us to move around and to manipulate the objects in our environment. Let me mention that this pathway, the blue one, the magnocellular dorsal pathway, is completely colorblind. It doesn't really care about wavelength information and color. However, the parvocellular pathway, which is the upper 4 pinkish layers of the LGN, does carry information about wavelength and local orientation, which is still kept completely separate in the top layer of the primary visual cortex still, and even in V2 and even in later areas like V4. So form and color are processed in separate pathways in low-level vision up to at least an intermediate area, which is called V4. And this is the information that I would like you to remember and take away from this slide because we are going to build upon this information later on. The separate processing of color and form in the brain, starting already at the retina, actually, is a very important feature of visual processing, and that's something that everybody who's doing scientific data visualization should keep in mind. Let me show you why. I'm bringing you further examples. This is to illustrate what I actually mean here. So this example shows you a nice old painting on the left-hand side from Sweden. The painting contains both color and luminance information, of course, so it very rarely occurs in nature or in art that a certain patch of color doesn't have luminance contrast, although we are not really aware of that. It's just natural to us. Now we know that color, of course, is defined by the wavelength of light, and color contrast is basically differences in the wavelength of light reflected from an object. Luminance, on the other hand, is related to the intensity of light reflected from an object. And these two attributes are really, as we mentioned, processed separately in the brain. Now, if we try to dissociate these two properties, luminance and color, what happens? Well, there is an image in black and white in the middle. It's not very surprising. If you Xerox copy any color images or just get rid of the saturation in a digital image processing software, then you're going to end up with this result. The black and white version is not bad. We can figure out the people in this image. We can sort of see the structure of the environment, see the trees, even figure out that there might be some sort of a shore, maybe it's a river, or is it the sea? It's not exactly clear, but it's not clear in the original image either. On the right-hand side, with some digital tricks, of course, because that's not particularly obvious, we only see color contrast. So all luminance differences between the colored surfaces disappeared in the right panel. So it's only color contrast. It's never perfect. Color at isoluminance might be near isoluminance for one person. It might have some luminance contrast for another person, because this is not exactly a physical isoluminance, it's a perceptual one. At isoluminance, it seems that we cannot really figure out any shapes. The structure of the environment is lost. Color basically only gives us the mood of the original painting, nothing else. Now, artists seem to be aware of this dissociation, actually. They use luminance contrast, which is the dark lines in this picture, to define form, while they treat color very freely, mostly providing large-scale shading and mood-defining information. The two, color and luminance, are normally, as I mentioned, together, and they complement each other and enhance the artistic experience. Scientists, on the other hand, are really main creatures, and they like to play with our senses. In this example, there is only luminance information at the maximum contrast. Although details are missing and it is mostly shading information that is presented, the face is easy to see on the left-hand side. The right panel has a reverse luminance polarity image. What was black is white and what was white is black, just like in a negative photo print. The face is kind of hard to see in negative luminance polarity, and this is because the visual system has learned that shadows should be dark and surfaces without shadows should be brighter. So it doesn't make sense in negative luminance polarity to our brain. This pair of images will be taken further to reveal to you what happens to form perception when luminance contrast disappears and there is only color contrast. Now we're going to try to build isoluminance. But since luminance contrast depends on how is the image projected, for example, what monitor at what setting is being used, and it also depends on the optical properties of the eye lens. For example, older people have yellower lenses. A little trickery needs to be done to make it sure that all viewers will experience what I'm attempting to show. Color at zero luminance contrast, that is isoluminance. The trickery starts with 3 pairs of images with equal lightness values for teal, magenta, and yellow and gray. The right panels have the easily seen positive luminance polarity versions, and the left panels, at least at the beginning, are the negative polarity versions. And here is a series of variations on contrast. The lightness or luminance of the colored parts of the double face images is varied. From row 1 to row 8, the colored parts are becoming lighter. The lightness of the gray areas remains at the same level in each line. The luminance of the colored surfaces is darker in the first row than the gray, and becomes darker than the gray when the double face flips and the left panel becomes an easily seen face. For teal, for example, in the first 2 rows, on my monitor at least, with my eyes, it seems that the flip is clear in the 4th row here. This is where the face is on the left-hand side, not on the right-hand side. The most interesting effect appears in the 3rd row for me, however. Look at it. I'm not sure what you see. Perhaps that occurs for you in a different row. But for me, it's strange. I cannot see the face in either panels. It seems that the gradation was good enough to reach exact isoluminance, at least for me, and on my monitor. And without luminance information, my visual system is not very good in form processing, as we know. Now, let me mention again that this is not physical equiluminance. If we had a photometer, it might show intensity differences between teal and gray in the 3rd row. And the visual system of a different person on a different monitor with different monitor settings might also observe the same effect in a different row. So this is a perceptual isoluminance that I can observe in the 3rd row, which leads to a temporary form blindness. Now check out magenta. The face is still on the right side in the 3rd row for me. So this is the positive luminance polarity face. It may be still on the right side in the 4th row. And if you go on, for me at least, it will only flip in the 6th row when the face is going to be on the left-hand side. And yellow might also provide a different result. Of course, this is because the color grants in your monitor and red and green are very different in this respect. The intensity is not equivalent to each other. So for different colors, isoluminance might occur at a different point. From this example, let's just take our first message. So we always have to remember that form perception is degraded at isoluminance. It might be useful to remember that different colors have different isoluminant levels with the background. Different people might also have different isoluminant points between two colors, and different image projections might change the isoluminant point between two colors. And this is very important information, I believe. But why is it so important? Why should we remember that? Well, naturally, if you use black and white, that's the maximal contrast, at least in print, on the monitor. In nature, it's a question, what's the maximal contrast? But we're talking about presentations anyway. If you add color, this is going to be at the expense of luminance contrast for sure. You will lose some luminance contrast by adding color. Adding some background, the luminance contrast will be reduced even more. And adding a pretty background to this pretty color might even send you into the isoluminant range, which is, it might be useful if you're trying to, let's say, generate an advertisement and try to draw the attention of people to your image, which they cannot really see. They cannot tell the forms apart, they cannot read it. So they go closer and they try to read it. And just by spending more time, more energy and attention on that picture, they might remember it better. However, when we're doing presentations, that's not the best thing to do. And the situation gets even more difficult when you reduce size. So at a small size, isoluminant text can be completely illegible, while black and white is still easy to read. Now, if you remember the simple fact that isoluminance is not good, you might actually do much better than some of the professional tools that we use nowadays to generate our presentations. This is, for example, a free PowerPoint template. Notice the very low letter contrast. It's pretty surprising, actually. For the B, there is almost no luminance contrast here. They provide a free YouTube demonstration how to use this template. And on the first slide of this free demonstration, we can notice even more than just low letter contrast. So for example, here, at this location, the contrast between red and green is probably gone in terms of luminance. If we take this luminance contrast, let's just take this luminance contrast with the same hues and apply it with our double face image, it seems that isoluminance is almost perfect. So I cannot really tell on which side is the face now, because I just cannot see the face. My form perception has fallen apart. And also in the lower right corner of this free demonstration, there is something written. Can you see it? Within the black circle, there is a text. It's not easy to read. However, if it was shown in black and white, right, like here, even in small print size, it's easy to read. The red and green contrast again is perhaps more perfect isoluminance than the previous one. Would you actually subscribe to that? I'm not sure. If you use the same contrast on a line graph that shows the impact of isoluminance on form perception, we're going to use this line graph later on in part 3 of the lecture. And please remember always to use high luminance contrast. It's very important. You can save the world, your mentors, thesis advisors, your future colleagues, and all other people from those terrible presentations with illegible letters and graphs where people get a headache because they are just trying to make sense of the letters that are in near isoluminant contrast. Please avoid that. And let's see what is the summary of part 2 of this lecture. We talked about the parallel processes of low-level vision to code for low-level features. We talked about the anatomically separate pathways. We mentioned that wavelength or color and orientation, that is form information, are independently coded in the parvocellular ventral pathway of the brain. We established that color information without proper luminance contrast does not really serve form perception. Form perception actually falls apart without luminance contrast. And we emphasized the importance of using luminance contrast at the same time with applying colors to avoid isoluminance. Thank you for your attention. This is the end of part 2 of the cognition lecture.